Hey everybody. Hope you're ready to start the next section. So from here on until the next exam, all right, the next exam is going to recover the next 3 weeks. Okay. First things first, just try to remember that quizzes and exams are due before that Friday at midnight. All right. So Friday, 11:59 p.m. That's going to be your last submission of ability. Okay. So my advice is to try and get it done early in the week so then you're not trying to rush and possibly miss the deadline. All right, so just take advantage of that. So moving on to the cardiovascular system, okay, this is going to be broken into 3 different weeks to talk about it. That's because there's 3 main parts of this system. There's the blood, the heart, and the blood vessels. This week we're going to focus on the blood. Okay, and so I'm sure you all know what blood is, correct? Blood is just a liquid connective tissue. I bet you didn't expect it was a connective tissue, but yes, it is considered a connective tissue. All right, it has a lot of stuff in it. It's not just your red blood cells. It does have red blood cells, but it has white blood cells. It has what are known as formed elements, cell fragments, and it has plasma, which I'm sure some of you have heard, plasma, blood plasma, which is considered liquid extracellular matrix. So, one of the key things you probably know what blood does is that it brings the oxygen to us. Okay, and we'll see a little more on how it does that, but blood helps to transport. It's one of the first jobs it does. Okay, oxygen, we breathe it in, gets into our circulation, and therefore the red blood cells bring it to the tissues that it needs to go to. Okay, so it's there to transport oxygen. Now on the flip side, when we're exhaling carbon dioxide, right? So that's considered waste. So blood helps us with the ability to take whatever carbon dioxide was in that tissue, let it circulate out into our lungs, and breathe out. Okay, so think of the blood kind of as a highway when it comes to oxygen and carbon dioxide with 2 cars on both sides. It also allows us to get our nutrients. Okay, so it helps with getting all the vitamins, everything we need from our gastrointestinal tract gets into our blood system and then expands out into whatever cells it needs to get to. As well as hormones. Okay, so we talked about the lymphoid system a little bit, and we talked about hormones and everything, and this is the main way hormones are going to get around. All right, so we're going to express those in our lymphoid system, our lymphatic system. It's going to get into circulation via blood, bring it to our targets. And then waste, we already said carbon dioxide. Okay, it helps with bringing out waste that has to go to the kidneys, waste that needs to go out of the skin and out. As well as, lo and behold, it brings heat. Okay. Not so much, I guess you can say, a physical transport. It's not carrying oxygen or carbon dioxide, but it helps to transport heat across and through our body. So basically, blood just connects everything, especially our respiratory, digestive, endocrine, urinary, and integumentary systems. All right, so it connects everything in our body. Now, its next function that it helps do is regulation. All right, so first one was transportation, and what it helps us regulate is our pH, okay, temperature, fluid balance, and hormonal regulation. So our normal range of pH and survival with our blood is about 7.35, 7.45. We'd expect that, right? Because we don't want our blood being extremely acidic, and we don't want it to be extremely basic. And if you remember little ways back, one of the ways that pH is regulated, all right, in chemistry and whatnot, is we use buffers. Okay, our blood uses buffers too, especially what is known as bicarbonate. All right, so it uses a buffer system to help regulate this pH in our body. All right, and I just said that it helps carry heat. All right, so it helps regulate our temperature. Plasma water absorbs heat. All right, so heat is literally just more of a thermokinetic type of energy. Okay, and our blood flow, if our blood is the speed of flow, you can say, to the skin and out is what helps us to regulate our heat loss, or how much we're holding the heat in. So our blood system, our plasma specifically, is there to help us regulate our temperature. Fluid balance, that makes pretty much sense because that's where a lot of our fluids go, right? You're drinking your electrolytes, all that fun stuff. And plasma proteins help to maintain osmotic pressure. So if you remember, probably in your previous classes, the osmotic pressure is pretty key with allowing basically the pressure of bringing the fluids through the veins as well as into the cells via going across the barriers of the plasma membranes and whatnot. Okay. And with it, like I said, osmotic pressure, it impacts water movement. Okay. So it impacts if it has the ability to flow between the blood and the interstitial fluid. All right. So it's helping there with hydration. I said it impacts the endocrine system. Okay. So obviously, one of the things it's going to do then is regulate our hormones. All right, specifically, three huge ones that it regulates is aldosterone, antidiuretic hormones, and atrial-neuritic peptides. And why those are huge is because those three themselves impact all those above. Okay, so each three of those are regulated by blood itself, but it also assists in regulating the three pH, temperature, and fluid balance discussed, hence antidiuretic hormone, right? You'd expect that to impact the water or the fluid balance, therefore. And its next job blood does for us is protection. All right, so blood is actually there to help a lot of our protection of bodily functions and, against any foreign invader. One of the main ones you probably know is obviously if we lose our blood, we need to stop. So we have clotting. Right? So it helps protect us from losing too much blood or hemorrhaging by clotting. And it helps by letting the spread of what are known as white blood cells. And we talked about that a couple weeks ago. Our macrophages, our T cells, our B cells, all those guys, it helps with flowing them through our bodies. And with the side of that, we talked about antibodies being produced by our immune systems. So those antibodies that are being produced by the immune cells are also able to flow through with the blood to whatever location of an invader or damage it needs to go to. It also helps with complement proteins and interferons, okay, which we've seen before that they help to interact with the white blood cells, tissue damage, activation of the complement pathway. So you can see a lot of the protection basically comes from its impact with our immune system as well. All right, so physical characteristics of blood. All right, so it's not like blood can do its own willy-nilly thing. It's got to be pretty steady across the board just because, it's that important for us, obviously. Temperature has to remain steady, so it's commonly about 38 degrees Celsius or 100 degrees Fahrenheit. I said our pH is very neutral, a little bit basic, but that's where that buffer system comes from to help regulating. There's some variation between sexes with volume. Males normally a little more compared to females. And about 8% of your body weight is coming from the blood within your system. Okay. And obviously, if you've ever cut yourself, you've seen color-wise that it is bright red. Okay. However, its color changes with its exposure to certain chemicals. And by certain chemicals, I'm talking about oxygen. All right. So our blood is bright red when it's oxygenated. Right, so when it has oxygen. It changes into more of a darker red color when it's deoxygenated. So there's no longer any oxygen associated with that red blood cell, for instance. Okay. Blood is much denser than water. Okay. It's more viscous and it's slightly sticky. Okay. So that's why it's if you cut yourself, it's a little thicker, it's a little stickier when it comes into contact with your skin. It's harder to get off a little bit. So, I talked a little bit in the beginning that blood is more than just red blood cells. All right, and you've probably heard of blood plasma. All right, but it's also made a decent amount of formed elements. Okay. So talking about plasma first, plasma's composition, it is mostly water. Okay, it's mostly water. It has some solutes in it. About 8.5% of it is going to be your solutes, and then 7% of that are proteins. Now, there's three big proteins that we have that are in our circulation of plasma. Okay, albumin, globulins, and fibrinogens. Each having their own functions. Okay, so they help with osmotic pressure, transporting, as well as immunity and clotting. Okay, each has their own role, and we'll get to what exactly they do. Now, besides those proteins, the other solutes are your electrolytes, whatever nutrients that may be taken in, hormones, gases such as our CO2 and our oxygen, waste, so urea, for instance, stuff that needs to get out of our body. All right, and plasma, as I'm sure you've ever heard of plasma transfusions, it's got a pretty decent clinical link. Okay, and for instance, edemas. Okay, it's an impact on plasma proteins. Okay, so we have albumin, which impacts osmotic pressure. This, if you have low albumin, you have a loss of oncotic pressure. So it is considered a classification of edema. All right, so plasma and its solutes itself can strongly impact our health. Now, formed elements, those are our cells. Okay, and there's three of them. Now, each one has its own class, but overall, there's three. Red blood cells, or RBCs, white blood cells, or WBCs, and platelets. Red blood cells, we know they're really there to help do our oxygen transport, and they're basically 99% of all the formed elements we have. All right. White blood cells are a little less than 1%, and that's where we've discovered, neutrophils, eosinophils, basophils, monocytes, lymphocytes. We talked about all of these. So that is the other portion of formed elements. And lastly, we have what are known as platelets, okay, or cell fragments. These guys are huge when it comes to clotting and helping out with clotting. So this is just a quick little picture to put in your head exactly what I'm talking about when I'm saying these elements or these cells. Okay, our nice lovely 3D picture of our red blood cells, exactly what you would expect. You've seen photos before. Our platelet cells, and then our white blood cells. All right, as well as if you want to see it more on a blood smear, this is what you would see. And now, most importantly, and what to remember is that our red blood cells do not have a nucleus. So that is why if you're looking at the blood smear on the right, you'll see some cells that have nucleus, okay, such as a leukocyte, as well as you'll see these guys don't. Okay, this is where the concave portion of the red blood cells come from. And of course, platelets as well, not so much. Okay, so they're much smaller when it comes to our white blood cells and our red blood cells, if you'd ever look at it on a smear. So, this again has pretty strong clinical relevance. All right, so our portion of plasma and our formed elements, they have what are known as a hematocrit. All right, so that's just the percentage of our blood volume that's composed of the RBCs. All right, normally each sex has its own little range, 38 to 46 for females, 40 to 54 for males. Okay, and you'll realize it's pretty high in males. So the real question is, why is it so much higher? And that comes into more of hormones. Okay, males have higher levels of testosterone, as I'm sure you know, and the higher levels of testosterone lead to an activation and cascade of a pathway, okay, and productions of proteins that are associated with an increased production of red blood cells. So males will normally have a higher percentage. Now, if this hematocrit is impacted, say it's low, so low percentage of RBCs, we have an anemia. So we have a reduced oxygen delivery, which makes sense because we don't have as many red blood cells to go do the job. And there's a lot of ways this can be caused. Okay, iron deficiency, for instance. We need iron in our blood to help with the transport of oxygen. Kidney disease, dramatic blood loss. Women are very susceptible to anemia, especially when it comes from menstruation bleeding, associated with the iron deficiency or the blood loss as such. And the other one is if we have it, I want to say too low, but kind of higher. Okay. Polycythemia is more of its effect is it makes your blood thicker. Your blood pressure is much higher. You have an increase of stroke risk. And what really can cause that is, if you're in an environment of hypoxia, dehydration, if you are blood doping, it can cause these kind of side effects. Now, this is just a way for you guys to visualize exactly what I'm meaning when I'm talking about blood plasma, RBCs, all that fun stuff. Okay, so if you're looking at A, now this, imagine, I go forth, I collect a milliliter blood, and I centrifuge it. What happens when you centrifuge it is it's going to separate, and it's all going to separate by weight, okay, and technically the viscosity of the liquid as well. And what you see is we have higher blood plasma as expected. Red blood cells a little lower. And then we have this little tiny section you'll see that's kind of in between clear and foggy. If you ever looked at it in a vial yourself, that is what's composed of white blood cells and the platelets. It's known as the Buffy coat. Okay. And so if I was to take that and I looked at it a little further in our body weight, all right, you would see that again, 92% of us is just other fluids and tissues, and 8% of us is whole blood. Okay, so the whole entire blood. Now, if I broke it out into those two sections, blood plasma versus formed elements, we'll see that, again, blood plasma is a majority. Blood plasma is highly water, but it has these key proteins which you should remember, as well as our electrolytes, nutrients, gases, waste products, all that fun stuff. Now, if we're at the formed elements, we see that kind of like the blood plasma, the highest amount of formed elements is red blood cells, which makes a lot of sense. Okay, and then from there, one of the smallest amounts is white blood cells, which breaks off into our immune cells that we discussed about before. Okay, so this is a good little kind of chart to help you guys study and understand exactly what's in what and how much is in such. Okay, so homeostasis. Okay, so we have to form blood cells, right? Or it's going to end up very badly. And the first thing we need to know is where exactly this happens. Okay, and it actually changes over our development and over our age. Okay, so before birth, it's your yolk sac, it's the liver, the spleen, the thymus of the developing fetus. After birth, it's your red bone marrow. Okay, and when you get to adulthood, it's axial skeleton, pelvis, sternum, proximal femur, and humerus. All right, so key thing here is just that it changes over time, location, where the formation of it is occurring. And what this is all coming from in those regions is known as the stem cell hierarchy. And I'm sure you've all heard of stem cells, those cells that have the ability to differentiate or turn into whatever cell they want. I'm sure you've heard of stem cell therapy. Our blood comes from this idea of stem cell hierarchy. Okay, so multipotent stem cells, or homeocytoblasts, they develop in these regions, okay, that we just discussed, into two major lines. Right, so two major lines of stem cells. One is the myeloid stem cell, and the other one is the lymphoid stem cell. Myeloid stem cell is strictly in a red bone marrow. All right, and that goes and gives rise to your red blood cells, your platelets, neutrophils, eosinophils, basophils, monocytes, so some of the more immune cells. All right. And now these guys can also differentiate into what are known as progenitor cells. Okay, so it is basically not quite to where it needs to be or the action it needs to be, and it commits to making whatever specific element of the blood is needed. Now, the other one, lymphoid stem cell, that goes from red bone marrow, okay, but the final, I guess you can say, active stage occurs when it gets to lymphoid tissue. So as such, lymphatic, lymphoid, that's our B cells, T cells, and our NK cells that we discussed a while ago. So the key thing to remember here is myeloid stem cell, one region doesn't move. Lymphoid stem cell, it's in red bone marrow. It moves through our circulation into lymphoid tissue. Okay, so remember that as the two differences. And each conversion to whatever you need to get to is determined by what are known as growth factors. Okay, so factors to determine the growth of the cells. Okay, and they each are produced by certain regions. They each stimulate certain things. EPO is produced by our kidneys specifically. It helps to stimulate RBC production. So if you remember, that is one of the key players that we talked about in testosterone in males, and it leads to red blood cell production. So that is a key growth factor in allowing to support the red blood cell production. And what occurs is it's very low in kidney failure. Okay, so clinically, if you have kidney failure, it's not really able to make these growth factors. All right, so as such, as a result, you're at higher risk or diagnosed with anemia as well, because you're just not making this factor. TPO is produced by our liver, okay, and that's there to help stimulate platelet function. You'll see down the line of this lecture that that's key with helping with our clotting. Colony stimulating factor, CSFs, and interleukins help with stimulating white blood cell production, which we saw and talked about in our lymphoid systems. Okay, so those guys are there to help with that. And I know we just talked about this, clinical applications, getting our anemia from the kidneys. But CSFs, for instance, they can be applied to patients receiving chemotherapy, especially those that may have their white blood cells being impacted strongly or don't have such a strong immune system, and allowing this application of this growth factor to promote their white blood cell production. Okay. Now this is a really huge hierarchy of how this occurs. Okay. So it all starts with a multipotent stem cell. That is just a stem cell that can go multiple different ways, hence its name. And in this instance, this is where the division, I said, of those two, myeloid stem cell, lymphoid stem cell. Okay, lymphoid stem cells, all right here. And what these are the precursors. Okay, so they don't just go strictly from, the stem cell straight to a B cell. All right, so what happens is they make some precursor cells, all right, for both the lymphoid and the myeloid stem cells. Looking at the lymphoid stem cells, these are just lymphoblasts, starting with our TB or NK. These guys get certain signals, and they go forth into T to B to N cells. Now, the myeloid stem cells, these guys need a signal beforehand to say, I need to become this precursor. So that is when these compounds or these proteins known as colony forming units, either erythrocyte, megakaryocyte, or granulocyte macrophages come in, and we'll talk about each of those, and they generate a progenitor cell as a response, and then these progenitor cells then are able to become more of the precursor side of things. Okay, so remember, multipotent stem cell, myeloid stem cell, they make progenitor cells on the myeloid stem cell part. Okay, and those progenitor cells then undergo whatever signaling pathway they need to determine which exact one they're going to make. Okay, you don't have this on the lymphoid stem cells. Okay, not so much progenitor cells on this side. Now, when these guys get the progenitor, they go through the precursor cells. They can go forth through the precursor cells to, eosinophils, basophils, platelets, red blood cells. And what you'll see when we get to more of our red blood cells and platelets is there's a little step that happens in between. Okay. One being nucleus ejection. That occurs at what is known as your reticulocyte. And we'll get into more details on these. And that is when, literally as the name says, nucleus is ejected and our red blood cells form with no longer a red nucleus. Okay. And some of them will end at this point. Eosinophils made, a basophils made, so forth, so forth, so forth. Some of them will go past to more of a tissue cell. Okay. These guys in the orange are considered circulating cells. A few of these guys will go forth into what are known as tissue cells. So mast cells, for instance, from the myeloid stem cell side. Monocytes become macrophages once they're in our tissue. B lymphocytes become plasmacytes. Okay, so the good takeaway here is that one, we come from the stem cell into two divisions. Myeloid stem cell uses progenitor cells, okay, all going into a circulating cell population with some continue on to differentiating into a tissue cell. All right, no, that was like super a lot. Stem cells, fun, right? But we're going to look at each one of them a little more and exactly how they're made. But first, we're going to start with red blood cells. I feel like if there's anyone you probably know a good decent amount about, it's probably these guys. And again, red blood cell count is dependent on a lot of things. Okay, and a lot of it, is just dependent on the biology of the person. It can also depend on sex. So male has a little more, female has a little less. Again, that ties back to the testosterone and all that fun stuff. But our production rate is about the same, and that's about 2 million per second. That's if you're a healthy human, not a diseased. So about 1 second every single breath, you're making 2 million new red blood cells. Okay. Now, these red blood cells have a certain characteristic, and like I said, one of them, the big one, is they don't have a nucleus. Halfway through the making of red blood cells, they shoot it out. Now, because of this lack of nucleus, it kind of gives this concave. All right, so it looks like a biconcave disc. All right, kind of like a little frisbee in a sense, as you can see on the RBC shape side. And what it's packed with is hemoglobin, which I'm definitely sure you guys have all heard about. And so hemoglobin has a specific molecular structure. Okay, it has 4 globulin chains, 2 alpha, 2 beta. All right, and then each heme contains about an iron. Okay, and each binds about 4 oxygens. All right, so it's an oxygen binding protein. So this is the protein that's in our blood cells that allows us to grab onto the oxygen. And they always will contain an iron, and that's why iron is so important. Because if you have an iron deficiency, you're not giving your blood the ability to grab the oxygen, to get the oxygen to wherever it's got to go. Okay, and this hemoglobin with the iron, and it's really what gives it its red color, and that's why when oxygen binds to it, the structure will change, and that will determine if it's dark red, if it's light red. Okay, so that is just the general characteristics and how oxygen kind of gets going. So the big takeaway I would say there is understanding what hemoglobin is, that it contains iron, that it's there to bind to 4 oxygens, and you need the iron to get its function, and that the oxygen binding and the iron helps to identify what the color is or give its color. Now on the flip side, besides oxygen, is the transportation of carbon dioxide. All right, and carbon dioxide isn't always transported by blood. It's got different ways to go. All right. A good portion of it is transferred as what is known as bicarbonate, all right, HCO3 negative. All right, that bicarbonate is done by blood plasma. Only 23% takes advantage of the hemoglobin. All right, so only 23% of that actually is going to bind to the hemoglobin and then go out. 7% of that is actually dissolved in plasma. All right, now the way to get it into bicarbonate is a nice little fun enzymatic reaction that we have to have occur in our systems. Okay, and that is where we use a specific enzyme known as carbonic anhydrase. And what occurs is the carbon dioxide in the water within our system comes together with the enzyme. We make carbonic acid, and in the process, carbonic acid splits. Okay, again, more of, I guess you can say, a little bit assistant of the pH control. It splits into a hydrogen ion and a bicarbonate ion. All right, so that's how it's transported through blood plasma. Whereas when it's bound to hemoglobin, it's strictly more of the CO2 portion of it, not so much a bicarbonate ion. So as I said, every 2 million for 1 second you get a red blood cell production. All right, but they actually stay in your system for a decent amount of time. All right, about 120 days. Again, this is if you're a healthy individual. Now, their lifespan lasts that long, and eventually, they will need to be either broken down or they will be destroyed themselves. And the lifespan really depends, like I said, on disease stat, but it just gets general wear and tear. Okay, your blood is circulating, it's squeezing through capillaries, it's struggling to synthesize any new components since it doesn't have a nucleus, so it just gets a lot of wear and tear over time. All right, so that's why the lifespan is, I would say, somewhat short than what you would expect, because they become more likely to just pop or burst. All right, so that can actually be pretty hurtful to us, if we just waste this red blood cell. Okay, so what occurs is that the red blood cells, one, they're removed from circulation. All right, and they're destroyed by phagocytic macrophages. So if you remember, those are macrophages that allowed for the internalization of whatever that it's going to destroy. Okay, so it's red blood cells at the end of its lifespan. It gets into a spleen and liver. Phagocytic macrophages are there. They help with the breakdown. Okay, so they engulf it, break it down. All right, but what happens is your body likes to optimize stuff as much as possible, especially to try and save energy. And if you're making proteins, you are using a lot of energy, especially complex ones. So to save that, they recycle the hemoglobin. Okay, not only do they recycle that actually, but they recycle the iron. Okay, so by breaking down this red blood cell, yes, we've had a breakdown of the proteins, some of them excreted in bile, but the main really important portions, the hemoglobin, iron, are going to be reused at the end of its cell's life cycle. All right, so scary picture, I know, but it will help you understand the life cycle. Okay, so we're looking at the life cycle of a red blood cell from time of death to new and rejuvenated in the full cycle over and over again. Okay, so if you're looking at number 1 on the far left here, this is when it starts with the death. Okay, so the macrophages in the spleen or liver, they engulf either a full red blood cell that's weakened or a ruptured red blood cell under the phagocytosis. Now, step 2, globin, which is a protein, all right, it gets taken and it's broken down and turned into amino acids. Now, when it's turned into amino acids, it can then go out and be reused for any type of protein synthesis that may need to be done. All right, so then heme, or portions of the hemoglobin, are split apart. And with that, if you remember, hemoglobin always has iron, okay, because it helps it do its job. The iron is also removed from that portion. And then that form of iron associates with a specific plasma protein. Okay, it's called transferrin. Transferrin acts as a transporter within the bloodstream for that form of iron. Now, eventually, iron makes its way with the transporter into muscle fibers, liver cells, or macrophages of the spleen in the liver. And it does that, iron will then detach from transferrin and attaches to an iron storage protein. That one's called ferritin. Okay, so it's acting as more of a storage method at this point. And now, at the time of this release from a storage site, say, it's ready to be used or recycled. Okay, it gets detached from that storage site or storage protein. It is absorbed from the digestive canal and reattaches again to the same transport protein, transferrin. Now it forms a complex, they're bound together, and it's carried to the red bone marrow. All right, so in the red bone marrow, that is where red blood cell precursor cells take it up through a receptor-mediated endocytosis. So if you remember, endocytosis is literally just entering, and cytosis taking in whatever it wants. Just at this point, it's led by a receptor. Okay, so it undergoes receptor-mediated endocytosis, and it brings the iron into its hemoglobin, and it's a part of its hemoglobin synthesis. Okay, and it's needed specifically for the heme portion. And the amino acids from the globulin portion that all the way back up broken down, those come into play with vitamin B12, and they synthesize the rest of it. Now, after that's gone into play, erythropoiesis in red bone marrow occurs. All right, it results in the production of the red blood cells, and now they enter into circulation. So once it's into circulation, when iron is removed from the heme, the non-iron portion becomes converted to biliverdin, so that's a green pigment, and then into bilirubin, which is a yellow-orange pigment. Now, when bilirubin enters the blood, it is then transported to the liver. All right, so within the liver, bilirubin is released by the cells into bile. All right, and that passes into the small intestine and then into the large intestine. Once it's into the large intestine, bacteria start to break down that bilirubin. All right, that came as a waste product. All right, so it starts to break it down and converts it into urobilinogen. So some of that is absorbed back into the blood. It's converted to a yellow pigment, and we just pee it out. But most of it is just eliminated in our feces, and that's what's a form of a brown pigment. All right, so gives it its characteristic color. Okay, so as you can see, we have our strict portion, so 1 through 8, that gets us to making a red blood cell, and then we have 9 through 13, which is showing how we get rid of that not so much used leftovers from the red blood cell when it's broken down. All right, so the production of red blood cells. So as you can see in the last slide, it occurs in the red bone marrow. Okay, and within the red bone marrow, as it's going through the process we kind of saw before, when the heme is getting there and the iron's getting there, it forms a proerythroblast. Okay, and proerythroblast divides into cells that help to synthesize hemoglobin. All right, and now this is all stimulated by that EPO. All right, so way back, and I've said it like 20 times, remember this is the growth factor that's impacted by testosterone, so it plays a really key role with red blood cell production, as you can see. So what occurs is the EPO is released, it then interacts with the proerythroblast, and it helps with production of what are known as reticulocytes. Okay, so reticulocytes is when they pop out that nucleus. Okay, so it has no nucleus, and it begins to enter into circulation. All right, so it's got rid of the nucleus, but it still has some other functions of organelles that are left over, such as like mitochondria, the ribosomes, the ER. So it's not like a completely hollow space of the cell, but it does lose its nucleus. All right. And now, once these guys are entered into circulation, and in about 1 to 2 days after they're there, they become official red blood cells. They've gone through the changes that they need to be. Okay, so it occurs in the red bone marrow, proerythroblast. They divide into cells that synthesize the hemoglobin. This is all stimulated by EPO. All right, works with those stem cells we saw previously. And in the process of it, it forms the reticulocytes. The reticulocytes then go into circulation, and that is what eventually comes to red blood cells. So, as you've seen through all of these lectures so far, is that there's always some form of regulation. So obviously red blood cell production is going to have a form, because it's pretty important, right? It's going to affect our oxygen delivery, our waste getting rid of. So it's going to impact almost everything throughout our entire body. Okay, so when we're looking at a feedback, it works as a negative feedback when it comes to control. So let's say we get a stimulus. Okay, we have a condition where oxygen delivery is occurring. What occurs is there's receptors in your kidney cells. They help to detect the oxygen levels. They detect the oxygen levels. They either increase or decrease the EPO secretion into blood. EPO gets into the blood. What occurs is EPO eventually get to those proethroblasts that we talked about in the red bone marrow, and they mature into reticulocytes. And that's when reticulocytes go and they enter the circulating blood, eventually becoming red blood cells. Okay, so that is the normal pathway, right? Especially if they get a response that's like, hey, we need to get going on blood production. Maybe, you got a cut and you lost some blood cells, or, you're in a region of hypoxia. But all in all, this stimulus gets to the point where you have increased oxygen delivery to tissues. Okay. Now, what can happen is you get some type of negative feedback, all right, where you need to stop this increased production of red blood cells. So negative feedback being such as, low oxygen content, or enough oxygen content. Getting signals back from the kidney cells to say, hey, we have oxygen, we're good. You're wasting energy making red blood cells. Let's dial it back. So it goes all the way back, stops the release of EPO, stops the production of the proetherblast, and so forth, controlling the red blood cell production. Okay, so the other cells, white blood cells. Now, for every about microliter of blood you got 5 to 10,000 of these. Now, I won't go like super in the details of this, because a lot of this stuff we covered way back when we did the immune stuff, the inflammation. But I'll go over kind of stuff that correlates to what's going on in blood. Okay, and so like blood, or red blood cells, we had to make them and we had to break them down, especially over wear and tear. So there are forms for that for white blood cells. We have leukocytosis, which is a response that occurs again from hormone responses or protein responses that increase your white blood cells. All right, so if you get some signal that there's an infection. Leukopenia, which is actually breaking down or lowering or feedback decreasing the white blood cell production. All right, so infection's gone. You've had a strong response to an allergy attack. You've now controlled the allergy attack, so it's the body's way of saying, okay, we don't need anymore. It's control feedback. But as we know, this is all to help defend against infection, whether it's internal or external. And each one of these have what we talked about as the MHC antigens. Okay, so all these guys are made in this process will have an MHC antigen, okay, or a cell identifier marker. And so when it's going through leukocytosis, you're making white blood cells, they're getting their MHC antigens. They either get classified as a granular or agranular. All right, and that classification depends on if you're looking at them under a light microscope, if they contain a chemically filled cytoplasmic granule that you can actually see under the microscope, or if they're not. All right, very scientific, right? So those guys we've talked about, right? Granular leukocytes, those are your neutrophils, your eosinophils, and your basophils. Neutrophils, most abundant, right? The nucleus has anywhere from 2 to 5 lobes, okay? And as the cell ages, the number of nuclear lobes will increase. So if you see the photo there, you can see there's several lobes, all right? The dark purple colors, okay? And if they start to age and they start to get more lobes and more lobes, they become polymorphonicular leukocytes, or PMNs, okay? Now for these guys, granulars are much smaller, okay, than those of the granular leukocytes. You can kind of see them. They're like little purpley type things there. They look like little pale granules. These guys don't strongly attract either acidic or basic stains, so that's why you don't really get them as prevalent to be able to see. Okay, so they're neutral loving, hence their name neutrophils when it comes to identifying them. And we have so much of these because they're the first responders. All right, if you remember talking about infections, they are the first ones on site to identify and begin the cleanup of whatever infection or damage. All right, so they're also there for phagocytosis. They're the ones that release their lysosomes, any reactive oxygen species. Like I said, stuff that's there to help with the initial attack or cleanup on an infection. Eosinophils and basophils. Okay, so on the left of this image, you can see eosinophils. These guys are really good for our parasites, allergies, histamine breakdowns with it. They have very uniform sized granules. Okay, they stain about red-orange because they like acidic dyes, so you can kind of see I guess I would say that's red orange, but they like acidic dyes, okay, and they usually do not cover or obscure the nucleus, and the nucleus will most often have 2 lobes. All right, so when you're looking at a basophil, though, which is on the right here, this guy, instead of breaking down, it releases histamine, it promotes inflammation, and it's much similar to what are known as mast cells, which we covered a couple weeks back. Okay, and these guys, the granules, stain blue-purple. All right, so they like the basic dyes. All right, so the granules will commonly actually obscure the nucleus. All right, so you'll have a hard time seeing the nucleus if you're looking at basophils versus our eosinophils. All right, so as you can see here, they use this acidic and basic dyes to help. Neutrophils, you had a hard time seeing them because they're neutral. They don't really care too much for the acidic or basic staining. Eosins, they love the acidic, all right, so they get like a red-orange color. And then basophils, they like the basic dyes, okay, so they look a little more dark purplish. Agranular leukocytes, all right, so these are probably more of what you think of when you hear, immune cells. So these are like your lymphocytes, your monocytes. Lymphocytes, they have a much longer lifespan, and they again have a fancy way of determining what they are, if they're large or if they're small, and it's all dependent on the measurement of size by a light microscope. So anywhere 10 to 14 micrometers is a large lymphocyte, 6 to 9 micrometers is a small lymphocyte. Now, a lymphocyte, you can see in these pictures, is on the left-hand side, okay, and the nucleus is pretty large, and it's definitely there. It will have a little bit of an indent on it, but it stains very strong with a slight rim around it. Okay, and these are your B cells, your T cells, your NK cells. So remember, B cells were the ones that made our antibodies. T cells were, again, really strong and important at killing infected cells or identifying infected cells. And NK cells were really like the surveillance type cells that would have a quick response to any cells that were either virally infected or cancerous. Now, monocytes on the right hand side, remember how I said that some of these cells go full activated once they get into tissue? These are monocytes. Okay, so monocytes, they become macrophages when they get into tissue. Okay, now macrophages aren't just created equal across the board. So if you remember, there was like pro-inflammatory, anti-inflammatory. But for here, when you're looking at it, you have what are known as resting macrophages or wandering macrophages. Resting macrophages, they're just in the tissue. All right, they're taking a rest from any type of inflammatory response. They're just, in a sense, guards waiting for something to occur. Now, if there's inflammation, it's a wandering macrophage because it's wandering the site of inflammation for any defense mechanism. Okay, and now when you're looking at it on the right hand side of this image, the nucleus is a kidney shape, okay, a kidney or like a horseshoe shape you'd see, and the cytoplasm is blue-gray, just like you would see as the other one, and it has this very fine granules kind of stuck throughout, so you can kind of see them as the little circle dots of purple. Those are the lysosomes, so that's what helps with kind of breaking down whatever the macrophages will take in. Okay, so white blood cell function. Again, we covered a lot of this before, but one of their huge ones they do is immigration. They need to leave the bloodstream, but the red blood cells need to stay in at the same time. All right, and they need to have some type of signal that says, hey, I have an infection. We have something over here that you need to come defend against, protect against, or impact. Okay, and so undergoing immigration, obviously it needs that signal, whether it be some type of cytokine or protein. It needs adhesion molecules, so helping it to stick into the endothelium of the vessels. It needs what are known as selectins and integrins, which help it with sticking, basically acting as adhesion molecules. And they need to complete chemotaxis and phagocytosis. Now, when you're looking at the immigration portion, it's pretty straightforward. Like I said, we covered this before. You have your blood flow, all right, and you have, for instance, here a neutrophil that's going on in. What occurs is you get some type of signal and says, hey, we have an infection over here. Now on the endothelial cell, you'll see they have some little adhesion molecules or selectins or integrins that are bind to. These rolling neutrophils or other immune cells, they will have basically complement proteins that will recognize these. Okay, and they recognize them and they act as a stick not only to them but into the endothelials as well. Okay, so you'll start to see them, they start to stick onto the wall of the endothelial cells. Now eventually they stick, and what they'll find is a space in between those endothelial cells, which are kind of laying over top each other staggered. So they'll find that space and they'll squeeze their way through it. Okay, and they squeeze their way through it, and that's when they get to whatever site they need to get to for a response. Okay, so that is the immigration portion. All right, and then we know they'll go forth, they'll go chemotaxis to get to where they need to be, to take in what they need to take, the phagocytosis, to then break it down with lysosomes and enzymes, if you remember that portion. White blood cells, though, it's not just like willy-nilly going off and doing whatever they got to do. They follow a specific pattern or traffic pattern. Okay, and it's all dependent on how fast they are, what they're responding to, what their job is. But neutrophils, they respond the quickest to any tissue destruction. Okay, and they respond to get there to do phagocytosis. They release a bunch of chemicals, lysozymes, oxidants, hydrogen peroxide, and they also contain what are known as defensins. So defensins are proteins that exhibit antibiotic activity, okay, and they release these vesicles that contain defensins, and it merges with phagosomes, okay. And now defensins, they're kind of like spears in a sense, and they basically poke holes in whatever infection micromembrane, and that basically causes it to leak. All right, and in the process, it leaks, it loses cellular content, and it kills the invader of the microbe. All right, so they kind of act as spears stabbing the microbe. So neutrophils are the quickest. They're the first ones to kind of get there. Now, eosinophils up next. Okay, they leave the capillaries and they enter into the tissue fluid wherever the signal response is coming from. Once they get there, they release enzymes such as histaminase, and that combats the effects of histamine or other substances involved in inflammation. Okay, and they help with phagocytase antigen antibody complexes. Okay, now after that, basophils will leave the capillaries. They enter the tissues, and those guys intensify the inflammatory response. So they release things like heparin and histamine, serotonin, all in granules to intensify whatever response. Mast cells, basically the same thing. All right, so they, similar to basophils, a lot of times kind of enter at the same time, a little after, but they are all involved in, again, in the similar inflammation. They release heparin, histamine as well, but this, as a difference to basophils, is mast cells introduce proteases, or enzymes that break down the proteins. Now, most lymphocytes will continually move among the lymphoid tissues, lymph plasma, and the blood, as we talked about prior. And they only spend a few hours at a time in the blood. Okay, B cells are particularly effective in destroying microbes and activating their toxics. T cells, we know they kill infected body cells. Natural killer cells are a wide variety of infected cells and certain tumors. So they're kind of just constantly in surveillance of it, and really their effect will depend on what exact signal of an infection that they're getting, what infection it is. All right, so however, while those lymphocytes are pretty quick to get into it because of the lymphoid tissue, monocytes take a lot longer time to reach the site of infection, but they come in large numbers, okay, compared to a small portion of the lymphocytes. All right, so monocytes, they will begin to enlarge, and those are guys, once they get there, they differentiate into wandering macrophages. So they enter the tissue as a monocyte, they then enlarge, differentiate into a wandering macrophage, and then the wandering macrophage, if you remember, it's active and it's ready to go through and either attack what it has to do, phagocytosis, or clean up the area of the infection. Okay, so those are the first two elements. Now we're getting into platelets. Okay, so these guys are also formed from the hematopoietic stem cells under the influence of hormones. Again, TBO helps with this guy. All right, so a pathway from those myeloid stem cells is myeloid stem cell, megakaryocytes, megakaryoblast. Okay, so if you remember, these guys had like a middle step cell, kind of like the red blood cells have. And what occurs is there's a transformation between that middle cell, the megakaryocytes, that causes cells to splinter into 2,000 to 3,000 fragments. Okay, each is enclosed by a plasma membrane. That becomes the platelets. Okay, platelets will break off the megakaryocytes. All right, in the red bone marrow. That's when they enter the blood circulation. Okay, so this whole myeloid stem cell to megakaryoblast, that is all done inside the red bone marrow. After the shattering occurs, that's when they leave. Okay, go into circulation. These guys only last about 5 to 9 days in our system. All right, and again, it's kind of similar to a little bit of red blood cell because they don't have a nucleus. They don't have a way to form new proteins, repair their wear and tear, and a lot of times they really help do some stuff that's important, so they're just used up really fast within our system. Okay, because these guys play a big role in clotting, for instance. So hence the function, it forms platelet plugs, it releases clotting chemicals. It's there to help promote any repair to your vessels, and it contains growth factors, okay, like we talked about prior. Now, this might be something you've heard about, platelet-rich plasma therapy. Okay, so Platelets really have tissue regenerating qualities. Okay, and so a long time ago, after some studies were done, they decided we could really use this for patients that have certain conditions like rotator cuff tears, tears in the ligaments, hamstring injuries, um, you know, skin regeneration, any type of wound healing. I think recently they started using it for male pattern baldness. I don't really quite understand how that one would work, but basically it's pretty straightforward. Okay, blood is drawn from patients. They collect the whole blood, they centrifuge it, they collect the platelet-rich blood plasma, and they inject. Okay, so they're kind of using it as, um, in a sense, assisting the plasma and the platelets to go to a directed, targeted area to help with repair. Okay, so since we talked about stem cells and kind of what we just hinted out a little bit there, of course, stem cell transplants were going to come up. Okay, because a lot of times stem cell transplants are used to address certain issues that come from not being able to use a lot of the red bone marrow and stuff like that. So there are two different types of stem cell transplants. One is a little more favored now, I believe, but they do both. Bone marrow transplant, which I'm sure you've all heard of, or hematopoietic stem cell transplants. Okay, and that is the replacement of abnormal red bone marrow with healthy stem cells. Okay, and it really, like I said, restores the norm of what you're missing. So it helps with rejuvenating red blood cells, so giving more oxygen, rejuvenating white blood cells, so more of an immune defense. If you have a clotting illness, it helps with improving the clotting, so platelets. There's also a difference in who's giving them. Okay, so there's autologous and allogenic transplants. So autologous is like they take marrow from you, okay? They then use the marrow they collected on yourself. And why this is very helpful is because it's got a lower risk of rejection. Okay, so it's kind of like a blood transfusion where there can be a form of rejection and it won't really work. You don't want to have that lovely needle done to you if it's not going to end well. So that's if it's your own. Now, allogenic, though, that is if it's not yours. Okay, so that's donor stem cells. All right, and that comes with a little more difficulty because there has to be tissue matching done. They have to do typing to make sure it's, good chance will be accepted by your body, and your body's not just going to attack it and fight it off. So as such, you have a much higher risk of having immune complications. Okay, so it comes more of what the condition is you're trying to treat, how typing may occur between donors and whatnot. So each has its plus, each has its downs. Okay, so looking at these guys, all right, so how they do the bone marrow transplant, it's a pretty straightforward harvesting procedure. All right, so typically they will collect from the iliac crest. Okay, so nice little, occasionally a butt cheek stab. You will be under general anesthesia, and it's pretty kind of straightforward. They use a large bore syringe and they dive on into more of the bone marrow region, so the red bone marrow, and they extract. Okay, and then once it is collected, if it has to go to another person, the typing is done. It is just injected intravenously into a recipient, just like you would for a blood transfusion. Okay, however, some extra stuff needs to occur, especially if it's coming from a donor. And before transplantation, patients have to undergo basically a reduction of their immune system, like chemotherapy or something like that. So that this way, if it's a donor transplant, they have a higher chance that their immune system's not going to just wreak havoc on exactly what they're hoping to treat. Now, the other type of stem cell transplants is contained from an umbilical cord after birth, actually. So this is usually the more favored one. A lot of people nowadays will actually um, collect umbilical cords after their child's birth, and you can actually basically store them forever, as long as they're stored under proper conditions. And then this way, if the child ever has a health issue or anything like that, then they have stem cells that are collected from that child to then do a treatment. And it's pretty easily collected, obviously with parental consent. It's got a lot of stem cells compared to doing what going into your hip bone would look like. Less concerns a lot with like transmitting infections or needing a specific type of match or anything like that. And it's, like I said, it's pretty straightforward. It's shortly after delivery. It is into the umbilical cord with a syringe. It's removed via the syringe, and obviously testing is done, and it's just frozen and stored. And they have basically blood banks for umbilical cord collection. Like I said, it's very big thing now with parents, just in case they need it for future child concerns. Okay, so blood, important, right? Need oxygen. So there's obviously an important regulation or homeostasis that comes with it. Now specifically, there's three mechanisms to keep this: vascular spasm, platelet plug formation, and clotting or coagulation. Vascular spasm is a smooth muscle contraction that is occurring. Okay, so if you look here on the picture on the right, it is literally just almost like a contraction flex of the smooth muscle around it, which is compressing connective tissue in the endothelial of an artery, which is either reducing the blood flow, or if you're releasing, increasing the blood flow, to help with reducing blood loss. Okay, and so this occurs when you're undergoing an injury, okay, cutting your hand or any other type of injury that maybe under the skin bleeding's occurring. It occurs with pain reflexes, or if you're exposed to specific chemicals, such as like ones that impact the platelet, because that would therefore impact the ability for the body to go clot and control the blood loss more. And you do it all over your body. It does happen everywhere, but there's a couple strong regions that happens a lot, and it's some of the most important: your heart, your brain, and your fingers and toes. Okay, and like I said, it's, if you think about it, you have cut yourself, okay, or you're bleeding, you have blood loss occurring. If you just let this water hose stay open and the water just keep going as normal, you're going to lose a lot of blood. However, if I take that water hose and I put a kink in it, or I squeeze it a little bit, it's going to reduce the blood flow. So in a sense, you're going to reduce the blood you may have lost. Okay, so it acts as a protection mechanism. Now the next one, platelet plug formation, acts in 4 steps: adhesion, release reaction, aggregation, and plug formation. It involves a lot of chemicals and a lot of basically cascade signaling. All right, so like clotting factors, ATP, calcium, thromboxane AT, which helps to strengthen a blood clot in a sense. Okay, serotonin, platelet-derived growth factor, which helps with proliferation of vascular endothelial cells, so helping with repairing damaged blood vessel walls. Now, starting with adhesion, step 1, okay, you got your blood vessel, your red blood cells, your platelets are going on by, and what has occurred is there's collagen fibers that are damaged, okay, and a damaged endothelium. So platelets will contact and stick to parts of the damaged blood vessels, especially collagen fibers. Collagen fibers is pretty sticky. Platelets themselves are sticky, but collagen is sticky as well. So they will see this tissue or these sticky parts and they will bind to it. Okay, so it's kind of almost at this moment like sticking tape over a rip in the paper. Okay. However, now the tape has decided to become active. And as the adhesion occurs, platelets do become activated. And their characteristics, they change quite dramatically. they extend projections, as you can see in the picture. They're no longer this tiny little, thing. They allows them to contact and interact with one another, and then they begin to liberate the contents of their vesicles, all while kind of basically activating other platelets as they're going by. So they're shooting out their vesicles in this process. This is all called the platelet release reaction. All right. And some of the things they push out is ADP and thromboxane A2. These guys play a huge role in activating those nearby platelets. All right. As well as serotonin and thromboxane A2 function as vasoconstrictors. All right. So they support the contraction of the vascular smooth muscle. All right. So decreasing the blood flow, just like we did with the first portion of homeostatic control that we saw. Now, next is platelet aggregation and plug formation. These guys basically go a little hand in hand with one another. And after the release of ADP, all right, other platelets in the area become more sticky and they start to stick. All right, and the stickiness of newly recruited and activated platelets causes them to basically adhere and originally activated platelets. They're kind of like making like a cluster, like a dog pile on one another. All right, and this gathering of the platelets is what's called the platelet aggregation. Now, eventually, as this aggregation occurs, the accumulation and attachment of all these huge numbers of platelets forms a mass that is referred to as a platelet plug. All right, so plug formation. So far we've seen controlling blood flow loss by constricting smooth muscle, and then we saw forming a platelet plug at a broken vessel to help with repairing, okay, and protecting any type of leakage. Next is coagulation or clotting. I'm sure you all heard of clotting and know what clotting is, okay? And again, if you've ever drawn blood or accidentally cut yourself, you know that blood changes its chemistry, not only just its color. And it will remain in liquid form if it stays within its vessels inside your body as it's flowing. But if it's drawn from the body, it starts to turn into a gel. It gets thick. Okay, so blood serum stays liquid. That is basically because blood plasma minus the clotting proteins. But when it starts to gel, it is forming a blood clot. And so basically insoluble protein fibers start to form within the blood, and that traps the elements of the blood. All right, so instead of this free flowing solution, it's basically developing this strong fiber that's kind of trapping it all together and getting it thick. Okay, so that whole process of those protein fibers trapping it, that's clotting. It's a whole cascade of enzymatic reactions. Okay, and it's there to fight thrombosis versus hemorrhage, loss of blood versus not enough blood. And it requires clotting factors. Okay, so we've said clotting factors so many times, but it requires clotting factors to do its job. It also requires calcium ions. Okay, so we have a lot of inactive enzymes throughout this process, especially in liver cells. And so calcium ions help a lot of times with enzymatic reactions with basically turning these inactive enzymes on. This also helps with being associated with platelets, assisting with release of damaged tissues, and the courses and identification. And if you ever see them listed, or if I'm talking about them, they are identified by Roman numerals that are indicated in their order of discovery. So you will see, if we start talking about them, that I may say, clotting factor 4 and then clotting factor 1. So don't be confused. It's the Roman numerals are done in order of their discovery, okay, not by the order of what they do. So clotting can be divided into 3 stages, okay, and the first step is basically the main difference, and that's because it can go 2 different pathways. It can either go the extrinsic pathway or the intrinsic pathway, but they all will lead to a formation of prothrombinase. All right, and this together, combined with one another, is the common pathway. Now, prothrombinase then converts prothrombin, a protein formed by the liver, into an enzyme known as thrombin. Thrombin then goes and converts what is known as soluble fibrinogen. All right, so that's again another protein formed by the liver into an insoluble fibrin. All right, fibrin forms the threads of a clot. Starting with the extrinsic pathway, this is a very fast pathway. Okay, it happens within seconds of any type of tissue trauma, and it's because it works with what is known as tissue factors. Specifically, this is thromboplastin. All right, and it leaks into blood from damaged cells outside of blood vessels. All right, it initiates the formation of what is known as prothrombinase. And now this occurs in the presence of calcium. All right, so calcium is in. This tissue factor begins a sequence of reactions and it activates a clotting factor 10. Once activated, it combines with factor 5 to form the active enzyme prothrombinase. And now that is completed the extrinsic pathway. Intrinsic pathway is much slower, so this takes several minutes. Okay, it's because it needs contact activation. So it needs activators that are in direct contact with either blood or contained with whatever damaged cells or anything. And so outside tissue damage is not needed. So that's one of the key differences. Okay, whereas extrinsic, we needed some type of damaged tissue on the outside. Okay, so what occurs in this pathway is endothelial cells, they either become roughened or damaged, so blood really can come in contact with collagen fibers, okay, surrounding connective tissue. And platelets are damaged, which leads to a release of phospholipids. Now with this exposed collagen fiber, this activates another clotting factor, okay, which begins a sequence of reactions that ends up activating clotting factor 10, just like what happened in extrinsic. Okay, and the platelet phospholipids, when it's in the presence of calcium, those also work together to impact the clotting factor 10. And now all that comes together, this clotting factor 10 is activated, it combines with factor 5 and it forms this active enzyme again. So you'll start to realize that these two pathways are very similar. Basically, the main difference is how it starts. But once it gets towards the end, they are basically identical. Okay, and now those come together and they form the common pathway. So after the extrinsic and the intrinsic pathway, prothrombinase is made. All right, and that marks the beginning of the common pathway. Now, a couple things happen in this pathway that are key down the road. One, prothrombin turns into thrombin. This is all done by prothrombinase enzyme, okay, in the presence of calcium, which helps catalyze that conversion. Fibrinogen becomes fibrin. All right, so thrombin in the presence of calcium, all right, so this conversion of thrombin first has to happen first, and then thrombin in the presence of calcium helps to convert fibrinogen to fibrin. So we go from soluble to insoluble. And now together, this factor 8 that is found within blood plasma helps to stabilize the clot that's forming. All right, so it's activated by thrombin and it helps to stabilize the fibrin thread specifically. Okay, so this factor is impacting both of them, but thrombin is really the key leader of this. Okay, once he is made, he can now follow up on those last two things. And so the common pathway, just like everything again, it's got regulation. Now, this instance, it's a positive feedback. Okay, now in the picture on the right, you see green arrows, and you'll see specifically there's a division of green arrows into two parts. Okay, our first positive feedback occurs with factor 5, and it accelerates the formation of prothrombinase. And so prothrombinase then in turn accelerates the production of more thrombin, and so on and so on. All right, and then the second positive feedback loop, thrombin activates platelets. Okay, and the platelets then go and reinforce the aggregation and further release of platelet phospholipids, which again goes forth and activates factor V. So the positive feedback from this common pathway will only impact intrinsic pathway, it does not impact the extrinsic, and it's in two places. Okay, so we got the plug formed. The whole goal was to limit blood loss. It's there, we have less blood loss. Now we undergo what is known as clot retraction, and then finally the repairing of the damage. Okay, so clot retraction is when there's a strengthening of the clot, and it's all done by tightening the fibrin threads. All right, so think of these threads kind of like you're throwing rope over something in the back of your car. You need to tie it down tighter and tighter. So what happens is the platelets contract them. All right, and it contracts them to the damaged cells very gradually. Now, as it's doing this contraction, what happens is vessels edges of the damaged edges, they become closer and closer and being pulled together. This decreases the risk of more damage, okay, but it also is kind of like closing the wound itself with the broken cells of the vessels. Now, as that continues, they get closer. We now do the repair portion where we have fibroblasts that are in the connective tissue come out. They either, refix the connective tissue, or they make new connective tissue, as well as incorporating new cells of the endothelial to help with vessel repair. But at the end of that, there should be a fixed repair. Okay, now this retraction really depends on the number of platelets that are in the clot. Okay, because those can impact the release of whatever factors that can assist with strengthening and stabilizing the clot. Now, it's not just platelet number that really impacts it or anything like that. There's also vitamin K that can really impact this. Now, vitamin K, while you may not think it's important, it's actually there and required for synthesis of 4 clotting factors. Okay, so with a vitamin K deficiency, it may impact your body's ability to make these clotting factors, and as such, limit its ability to do this entire process. So you might have blood loss issues. Obviously, clotting needs control. Okay, we only want clots to occur when they do. And actually, many times a day, little clots start to form. All right, it's often at times there's just like minor roughness, maybe a minor little injury, or developing blacks over time in a blood vessel. It just can happen. So we have to have ways to prevent any type of form of damage that can come from it. So when it comes to controlling it, it has a couple ways and a couple systems it can do it with. Fibroanalytic system, it is there to help dissolve small, just inappropriate clots, or clots at a site of a damage that basically is repaired. Okay, it's done its job, and it breaks down these clots that are called fibrinolysis. Okay, so one way. Now, also when a clot is formed, an inactive blood plasma enzyme, it's called plasminogen, is incorporated into a clot. Body tissues, blood contain substances that basically activate this enzyme. Okay, and it activates it to either plasmin or fibrinolysin, but it's an active plasma enzyme. Okay, and there's a couple other things like thrombin can activate it, TPA can activate, a couple activators can activate it, but no matter what, the whole endpoint there is it ends to an active plasma enzyme, and once it is active, plasmin is formed, it can start to dissolve this clot, and it does it by chewing away those threads, digesting those fibrin threads. And as such, it starts to inactivate substances such as the factors, the prothrombin, the fibrinogen. So it basically breaks apart the pathways as it starts to break apart the threads. Now, one of the other controls we want to have is we don't want a clot to just go beyond a wound site into our general circulation. That's not good. So our body has a couple ways of doing that. Some of them are just a natural side effect. One is the fibrin itself starts to absorb thrombin into the clot, which starts to limit the pathway. It's kind of like a feedback control. Another one is just that our blood is constantly flowing, right? We're constantly having this flow of liquid like a river. So there's no kind of traffic jam of factors that can help with clotting or clotting factors. Okay, so the concentrations remain just so low so that no widespread clotting can occur. And it's just a side effect of our blood flowing constantly, like a river. Another way is endothelial cells and white blood cells can actually help limit it. All right, they produce a prostaglandin that's called prostaglandin, and it inhibits thromboxane A2. Okay, it's a super strong inhibitor of platelet adhesion and release. So it would just skip the ability for the platelet to even start the process, to even grab onto the endothelial cells. And lastly, while you may think anticoagulants are just like a drug they give you, we produce them naturally in our body. And there's naturally circulating in our blood as a homeostatic control. We have antithrombin, which blocks several factors. Okay, and heparin, that is an anticoagulant that's actually produced by mast cells and basophils, and it combines with our antithrombin there and increases its effectiveness in blocking thrombin. All right. And activated protein C, it actually inactivates two major clotting factors, and it's not specifically blocked by antithrombin, but it enhances the activation of a plasminogen activator. So those enzymes that were utilized to basically break down the clots. No system is perfect. Sometimes blood clots will form within the cardiovascular system. It can happen from trauma, infection, just wear and tear over endothelial surfaces, but they can occur. All right, and sometimes this leads to blood flow that starts to slow, and these clotting factors can start to increase in concentrations, which allows the clotting to become even worse. But it's not good. Okay, and the clotting in an unbroken blood vessel, which is known as a thrombosis, it can lead to some pretty bad things because think about it, it's going to start impacting your blood flow, your blood pressure, the distributions of clotting factors, anticoagulants, even your white blood cells. So it's going to impact the ability to control infections by impacting the circulation. And a lot of them do dissolve kind of spontaneously, but sometimes it will remain intact. Okay, so it happens. It usually goes away. And if it stays intact and it becomes dislodged or falls off, it becomes what is known as an embolism. Okay, or just a piece of debris that's going through our bloodstream. And so if it breaks away from an arterial wall, it may lodge itself into a small diameter artery. Okay, and that's even worse. It's bad that it messes with our white blood cells and stuff like that, but this can just almost completely block blood flow to a vital organ. All right, pulmonary embolism is an example of this. Now, interestingly enough, aspirin kind of helps control this a little bit. Aspirin inhibits vasoconstriction as well as platelet aggregation, okay, because it blocks synthesis of thromboxane A2. And it also reduces chance of this type of formation, of thrombus formation. So due to these effects, aspirin actually reduces the risk of like strokes, myocardial infarctions, but it really kind of almost, I could say, acts as, I don't know, protective. I don't want to say go slug down a bunch of aspirin, but in a sense it can act in a moderate manner as a protective measure. Okay, so now we're getting into blood groups. This is something I'm sure all of you have heard or know of. I'm sure you all know your blood type. So blood groups are broken up into two major groups, ABO and RH. Okay, so these blood groups are determined by these genetically determined assortments of those of antigens on erythrocytes. Okay, and it's like glycoproteins, glycolipids. They're called agglutinogens, and they're based on presence or absence of various antigens that are in like a combination of one another. But it allows us then to organize into different blood groups. And within a blood group, there can be two or more different blood types, right? We have AB positive, AB negative, O positive, O negative. But there's at least 24 blood groups and more than 100 antigens that are detected on the surface of our red blood cells, which help us with understanding blood groups, assisting with transfusions, organ transplants. So it's very key for us to know what these groups are. Now, ABO, like I said, is probably one you guys are the most familiar with. Okay, and it's based on two specific glycolipid antigens called A and B. Type A is only the A antigen. Type B is only the B antigen. Now, if you're type AB, you're both antigen A and B on your blood cells. Type O, neither. You do not have an antigen A, you do not have an antigen B, as you can see here in the photo, as an example for the difference of them. Now, alongside with just the receptors or the antigens, apologies, on the blood, you have what are also known as antibodies called agglutinin, and they're within our blood plasma. Okay, and they react with either A or B antigens. So anti-A antibody for A, anti-B antibody for B. So type A, okay, is normally an anti-B antibody. Type B blood normally has an anti-A antibody. Type A and B doesn't have any, neither antibody. Type O has anti-A and anti-B. So knowing this and the differences of what's there, if the anti-A is there, if it really comes into play when you're doing transfusions. Okay, so I'm sure you all know just transfusions is transfer of blood components like blood plasma or blood cells into bloodstream or directly into a red bone marrow. Okay, and as we can see with A, B, C, D, E, F, G, all the blood types, something can occur. Our body maybe doesn't recognize type A blood or doesn't recognize the A antigen. So our immune system sees that as this is a foreign threat. I'm going to attack this. I'm going to destroy this. And in an instance, this is what leads to failure of transfusions. This is why understanding which blood type is which and how it can match up with one another is key. Okay, so they can trigger an antigen antibody response in a transfusion recipient depending on the combinations. Okay, and that is what causes incompatible blood transfusion. And that's when antibodies in the recipient's blood, so those receiving the transfusion, bind to the antigens on the donated RBCs, so red blood cells. Okay, so it does that, but if you remember looking at the antibody, it had several kind of little arms where it can bind to different antigens on the red blood cells. That can lead to a giant clumping because it's like an antibody that's grabbing onto 10 things at once and it just clumps it. So in a sense, we can't use the red blood cells and the immune system goes and attacks it. So that's when it comes into understanding which is which. Okay, because these complexes, they activate blood plasma proteins of complement family, making blood plasma membrane of donated RBCs leaky. Okay, so not only are they clumping together, but them coming together is activating blood plasma proteins that's coming in. It's basically poking holes inside the plasma membrane, and it's causing the rupturing of these blood cells. Okay, so it actually releases hemoglobin into the blood plasma. We don't get oxygen as a result of it, and there it doesn't work. Transfusion is not successful. So even worse, actually, is free hemoglobin can go and actually give us kidney damage. Or it actually can lead to clogging our filtration membrane. So not only does transfusion not work, you're not getting the repair of the blood volume or the immunity, but you can end up getting kidney damage or kidney disease at the end of it. So one of the downsides, but another side besides checking for antigens, things like that on blood, is that usually transfusions also come with checking of viruses, because while it's rare, some instances viruses like AIDS, hepatitis B and C can be transmitted through contaminated blood. So transfusions are very highly regulated, very highly checked for anything that could possibly go wrong. Nowadays, there's not much of incompatible failure. I think we've got it down pretty well to it. It's pretty much successful a good amount of time. This is just a quick example of what I mean with something goes wrong and it doesn't work. So I have a type A blood. The donor for the blood is type B. Okay, so A is receiving, B is donating. So the recipient's blood contains the A antigens, all right, on the red blood cells, and it contains anti-B antibodies in the blood plasma. The donor's blood, which is type B, contains B antigens and anti-A antibodies. So in this situation, two things can happen. First, our anti-B antibodies in the recipient's blood plasma will bind to the B antigens on the donor's red blood cells. This causes that clumping and the rupturing of the red blood cells. And second, the anti-A antibodies in the donor's blood plasma can then bind to A antigens on the recipient's red blood cell. This is not as serious as the previous one, okay, because the donor has anti-A antibodies and they become diluted in the recipient's blood plasma, so they don't really cause significant, rupture of the recipient's red blood cells, but it basically renders the transfusion kind of useless in a sense. It doesn't do the effect it's going to do. So when you're doing an incompatible transfusion, both red blood cell populations are being impacted. It's just that the recipient has enough blood and recovery to not so much impact its own RBCs. It's the donor that's being impacted the most. Now, I said A, B, O, all those, but I never said plus, negative, positive, all that fun stuff. That refers to what is known as the Rh blood group, and it's named by the antigen that was discovered in the blood of monkeys, rhesus monkeys to be exact. Now, these alleles of 3 genes, which are proposed, they code for this specific antigen. And some of us have it, some of us don't. O negative, O positive. I'm O positive, so I don't have A or B, but I have the Rh. So, normal blood plasma actually doesn't contain any anti-Rh antibodies. But transfusions are actually really difficult with its presence. Okay, so if I have an Rh negative person receiving an Rh positive blood transfusion, the immune system starts to make anti-Rh antibodies that remain in the blood. So if there's ever a second transfusion of Rh positive blood that's given down the road, those previously formed antibodies, because of the first exposure, those can then cause the clumping and the leakage. Okay, it's almost like you're giving your body a dose of vaccines and your body's making antibodies. It's just that it's making antibodies to something you want. Okay, and so Rh presence, it's literally just an antigen. So if you see the photo on the top right, we have A positive here. So A is there, Rh antigen is there as well. It just looks like another antigen. Still like figuring out kind of what it does, how important it is to us. But we know that it needs to be carefully watched when it comes to not only blood transfusions, but also babies. And I'll show you in a bit why that's important as well. But the first thing to say, I'm talking about transfusion and how, we need to know what's what and all that fun stuff. So what they do is called blood typing. This is how they determine what your blood was a long, long time ago. And it's a pretty easy standardized test. They're much easier now. This is pretty much old school way of doing it. But what they do for typing people's blood is they take just single drops of your blood and they mix it with different stains. Okay, and the different stains that contain antibodies. You got anti-A and anti-B. Okay, just like you would inside your body as well. 1 drop of blood gets mixed with an anti-A serum, and that contains the A antibodies. And then we watch the red blood cells that, if they possess the A antigens, will start to clump together, start to get leaky, all that fun stuff. And another drop is mixed with the anti-B serum, and it should do exactly the same thing, only if there's a B antigen present. Okay, and so if the red blood cells clump only when they're mixed with an anti-A serum, which by clumping I mean you can see up on the picture there, where you have untreated blood that's easily spread. On the other ones that are clumped, they're literally just clumped in little piles together. So if I see that this serum is there and it's only an anti-A that I have clumps, I know that I have a blood type A because only A has been clumped. Now, if I treat it with B and B is clumped, that means I know that the blood cells have B antigen on them because the clumping occurred. It's B. If I see both clumped, that means it's A and B because it has both antigens and it interacts with both antibodies. If I see neither, then it's O. They don't have A, they don't have B. And now a similar test is done for the RH. A lot of times some people will do it side by side with this. So it's easier to get a diagnosis that way. Now I'm just going to cover some disorders that you may know of, you may not know of. One of the one that kind of comes back to the RH and what I mentioned a little earlier is hemolytic disease of the newborn. And so it's basically the most common natural problem with incompatibility, and it arises during pregnancy. So normally during development, there's no direct contact between maternal fetal blood while the woman is pregnant, development, all that fun stuff. But towards the end, you can have leakage. All right, so you can have leakage of fetal and mother blood occurring. And if a small amount of, say, the baby is RH plus leaks into the mother may be RH negative, and she doesn't recognize RH plus, so she starts to make antibodies towards RH. And so what can occur is, if the mother doesn't get this special injection to control this, the immune system can start to attack the baby. Okay, we'll recognize the baby and its blood as a foreign object, and the immune system will start to attack. Okay, so this is why a lot of women, they suggest getting a specific antibody prevention, okay, that during pregnancy, usually after pregnancy as well, to basically dampen the mother's immune system from risking to attack. Okay, so my wife and I had to do this because she is negative and I am positive, and so we just had to do it as a precaution in case this would occur. Okay, so it's a very common thing. Now they got it pretty much under wraps, so you don't have much of a side effect occurring from here. Anemia, that's just our reduced oxygen carrying capacity. So it comes from a lot of stuff. It's not just probably one versus the other. You probably most commonly think of iron deficiency. So iron deficiency is one of the pretty good ones. Vitamin B12, so that actually will impact, the red bone marrow's ability to produce large or normal RBCs. They're usually large and abnormal. Hemorrhages, excessive loss through bleeding. Commonly, anemia is really common in women. It's because of the massive blood loss during menstruation, as well as during pregnancy and the blood that is utilized during pregnancy throughout. So next is we can just lose blood from rupturing. Right? If we're starting to lose our red blood cells, we're losing our oxygen capability. So if our blood cells are just prematurely rupturing or weaker, then we're going to lose oxygen capacity, as well as if our main source, the red bone marrow, is impacted as well. The second one you probably know the most is sickle cell disease. So this is just, instead of a normal hemoglobin, it's abnormal. So it doesn't really hold on to oxygen in a normal way that we would. And so this gives up oxygen, HS, to the interstitial fluid. It forms a different shape. Okay, so it becomes, instead of we see in the left there, a normal blood cell, it becomes a sickle shape, hence its name, a rod-like structure. Okay, and it bends into a sickle shape. And now this is bad because it can really lead to strong organ damage. It really impacts oxygen capabilities. Hemophilia, that is a clotting deficiency. So it's strongly genetically linked. It impacts males a lot because it's X-linked. So, males XY. So with most, X-linked diseases, we don't have much of a defense against them. Fun fact, it was actually considered the royal disease. A lot of males from descendants of Queen Victoria have it. So strong belief that it's just passed down through her chromosomes. But it's got a pretty decent treatment. I mean, lots of transfusions. You get treated with clotting factors to help control any issues it may have. Your only main concern is just watching for serious damage of excessive bleeding. The other is leukemia. Okay, so leukemia is unfortunately pretty common. It's cancer of the white blood cells. It's also considered red bone marrow cancers because that's where they come from. But it also impacts the production of red blood cells. So as such, it's going to impact your oxygen carrying. And leukemia comes as some different types, I would say. A lot of it is how it's derived and the basis of kind of symptoms. Okay, acute is leukemia that's come on pretty rapidly. Chronic is it's taken a long time for the leukemia to occur or the red bone marrow to be affected. Lymphoid versus myeloid is really dependent on the classification on the basis of the type of white blood cells that are actually becoming the malignant cancer. Okay, so lymphoblastic or lymphoid, it's all the lymphoid stem cells or lymphocytes. Myeloid is myelogenous leukemia. That is more of the myeloid stem cells, so like the myoblast. And common symptoms are anemia, infections, and bleeding. Everything that you would expect to come from an impact of red blood cell production or its ability to carry oxygen. Now, I believe that is it. Remember, quiz Friday before midnight. I will share the password and all the fun jazz. If you have any questions, just let me know. Feel free to swing by my office hours at any time.